On the other hand, we also have what we call genome editing or gene editing. Here, you can repair a defect using homology-directed repair. That means that you use gene editing tools in the presence of a DNA template that has the correction. On the other hand, there is also what we call gene knockdown and gene addition. Good examples of genome editing is the Casveggie treatment for sickle cell disease, whereby the BCL2A gene is suppressed in order to switch back to hemoglobin, fetal hemoglobin, in order to correct for sickle cell disease. For dual CAR T cells, these are T cells that have been genetically modified to express T receptors that are more robust at clearing HIV. They may also be CAR T cells for cancers. That means here there is gene addition, and we've seen it in practice for cancers. So what tools do we use for gene editing? We use the CRISPR-Cas9, and this is the one that is currently in the spotlight. The CRISPR-Cas9 is an RNA-guided DNA cutting nuclease. Basically, it's more of, we usually simplify it as a molecular pair of scissors. So this requires that it recognizes a particular site in order to prevent off-target cuts. And it can make double-stranded breaks in the DNA, allowing you either to add or to remove a section of the gene. We also have what we call TALENs. These are protein-based DNA binding nucleases. They have a high specificity. They do not require palm sites in order to work, but they are slow to design, they're expensive, and there are issues with delivery. So that's why people are moving away from the TALENs. Then we have the zinc finger nucleases, and these have a very high specificity, but the reason why we are moving away from zinc fingers is because they are hard to engineer and labor intensive. Then the new kids on the block are the base editors, which can convert a single base without causing the double-stranded breaks like the nucleases, the Cas9, the TALENs, and the zinc fingers. And this is more precise, and so you have fewer unintended mutations in case you use a base editor. A good example of a disease that would be cured using a base editor would be sickle cell disease, because for one to have sickle cell disease, it's just one singular change in the bases. And so if you can be able to cut and replace that, then you're able to treat for that disease. Then we also have prime editors, but they have a complex delivery mechanism. They have variable efficiency. That means you don't have guarantee of how it would work, and they are large. So if you also add the genetic cargo, that means that they are most likely not going to be taken up by the cells that you're targeting. So in terms of HIV cure, with the tools that we have, where do you target? How do you use those tools in order to achieve an HIV cure? So we look at the entire life cycle of HIV in a cell. We could target the CCR5, which is on the outside of the cell, which is on the cell surface, the co-receptor for entry. We could also target the viral RNA before it's converted to DNA within the cell. We could also target the viral DNA before it integrates within the nucleus. And also we can target the proviral DNA, all the actual viral molecules before they are released out by the cell. So this CRISPR-Cas9 can be used to cut the virus across the life cycle. And when the virus is defective, that means that you do not have infection. It cannot infect the next cells. Or if the virus cannot enter the cell, then viral replication cannot happen, and therefore you've cured for HIV. So CRISPR can also be used to knock out the CCR5 co-receptor. It can be used to delete parts of the viral genome. It can also be used to knock in genetic material. In this instance, if you want to do something like CAR T cells, that is, you're introducing a new gene, or broadly neutralizing antibodies, still you're introducing a new gene, and that is knocking. So basically, you're adding something to the cells. On the other hand, you can also activate the latently infected cells in order for them to go into replication. That's what we call the shock and kill strategy, whereby you can use CRISPR to activate the latent virus so that it can be exposed to other clearing agents such as antiretroviral therapy. So there you shock and then you kill using the ART. On the other hand, you can do what we call block and lock, whereby you use the gene editing tool in order to make the virus go into deep latency. And until that infected cell has reached its full life cycle and is cleared, then that virus will stay inactive until the cell dies off. And that way, with gradually, as more and more infected cells die off, the body is cleared of the virus. So these gene editing tools can be used across the life cycle of the virus. And when we talk of CRISPR, we talk of zinc fingers, we are talking of base editors. These are all small molecules, they are enzymes. The question is, how do we deliver them into the cells of interest? The number one delivery system for gene editing tools is viral vectors. Here you are looking at adeno-associated viruses. You're looking at lentiviral vectors, which are HIV-like. You're looking at also adenoviruses. And they have a very high efficiency because you're mimicking infection. Cells will take up the virus by virtue of what is expressed on itself on the surface of the virus. And however, there are challenges with using viral vectors because, for example, AAV, it has been shown that a number of individuals have been exposed to AAV. That means that if you use it, you're most likely going to get a very robust immune reaction to it. And therefore, either you need to use AAVs early on in life before exposure, or you use the alternative, which will be like the lentiviral vector. Then in terms of integration, lentiviral vectors are able to integrate, whereas AAVs do not integrate in the nuclei. Then when we look at scalability, viral vectors are very complex to manufacture. They're very complex to manufacture, but they are best for long-term expression of the gene of interest that you've transferred to the recipient. Then other delivery systems for gene editing tools are nanoparticles. These can be large, they are flexible. Usually there is no immunogenicity towards them, and they are usually non-integrating. They are easier to manufacture and scalable, and we've seen them in being used to deliver CRISPR and mRNA. Then we have electroporation, where you use electric shock in order to open up the cell membrane, and then the genetic material that you're introducing into the cell can be taken in. This is a non-integrating delivery system, and it's limited by cell handling because most of the cells would die during this gene modification. So when we look at gene editing, I've talked about the tools for gene editing. I've also talked about the different delivery mechanisms, but how do we achieve this within the laboratory or a clinical setting? So there are two main kinds of gene therapy: ex vivo, which means that the gene modification or the gene editing is done outside of the body, and then the in vivo gene therapy, whereby the modification is done within the body. And for ex vivo gene editing, you find that, let's say a person is living with HIV, so you collect their T cells, those are the peripheral blood mononuclear cells. Then you isolate out the cells of interest, do the gene editing either using CRISPR or zinc fingers in order to cut the maybe CCR5 or in order to cut the virus, and then you reinfuse back into the patient. And on the other hand, with in vivo gene therapy, basically the genetic material and the delivery system are simply put in a syringe, and then usually we target the muscular cells, and you give a shot, and then that is delivered to the muscle cells, and they would continuously produce the gene of interest. So manufacturing ex vivo cell and gene therapies is quite a long process. In the clinic, you need to screen the patient, whether they are eligible for this. If it is HIV cure, currently they are looking at individuals that are virally suppressed. If it's sickle cell disease, you're looking at an individual that does not have adverse organ damage from this sickle cell disease. So after screening the patient for eligibility, then you administer what we call pre-treatment. This is, you give mobilizing agents that would move the stem cells from the bone marrow into the periphery so that it can be collected by apheresis. Or alternatively, you can collect the bone marrow through surgery. And then in the laboratory, you go ahead and purify out the stem cells from this bone marrow. And here you're using immunomagnetic-based techniques. So you have beads that are made of either iron, and these are coated with antibodies that can bind to the stem cells, which are your cells of interest. Then when you isolate these stem cells, you can either cryopreserve them or continue on with the process directly, whereby you introduce your lentiviral vector or AAV, and then they will deliver the genetic material to modify the cells in a culture. This takes about 2 to 3 days, 2 to 4 days, and it depends on the target cells. If it's something like T cells, you would need to culture them further for like 10 days in order to expand the number of cells that have been genetically modified. Yet if it's stem cells, you need to maintain the stemness. You don't want them to differentiate, otherwise they would not go back to their niche, the bone marrow. So that culturing is just 2 to 4 days to allow the uptake of the delivery tools for gene therapy. Then after this culture period, the cells are are formulated in such a way that they can be reinfused back into the patient. That means you wash away the excess virus, you wash away the excess beads that were involved in the isolation process. You also do release testing, which I'm going to go into a little bit further down the way. And then when you know that the product is safe and okay, then you infuse back into the patient. But prior to that, there is need to still give conditioning in order to create space within the bone marrow where those cells are going to go. But also the conditioning would help to clear any damaged cells or infected cells. So that the cells that you're infusing have a better chance at engraftment. So from our work, we've seen our goal is to simplify that manufacturing workflow. And the question is, can we able to do this under 24 hours rather than weeks? So you would still screen for the patient, collect the stem cells, but we are looking at purifying these stem cells while also concurrently introducing the vector that is carrying the genetic material in order to modify these stem cells. And then you go under release testing and formulation of the modified cells and be able to give them back to the patient within the window of the pre-treatment, within the half-life of the pre-treatment drugs. So that way there is no need for conditioning because the pre-treatment drugs would have already vacated the bone marrow, which is the niche for these stem cells. And therefore, you cut on the time that is required to manufacture these gene therapies. So some of the release testing assays that are looked at before you give back these genetically modified cells, we look at sterility testing. There you do your different cultures, gram stains. Then we look at mycoplasma. We looked at the vector copy numbers. That is how much of the lentiviral vector is within the cells. Then we look at the bead content. That is how much of the beads that you use for isolation of these cells remains within the product at the end of the process. Then, of course, you look at purity, because the purer it is, the better. Then you look at if it's T cells, how many of them have now become CAR T cells. You look at in vitro cytotoxicity, cytokine secretion. Those are functional assays to see that these cells are actually functional. And then also you can look at persistency of these cells and also T cell expansion. So a laboratory that supports gene therapy would be able, for a cure for HIV, should be able to do viral load, CD4 counts, should be able to measure the HIV reservoir, should be able to look at renal and liver functions, and also it should be able to look at it should also be able to look at anti-drug antibody testing, and then transcriptomic analysis. So when we talk about gene therapy, sometimes it sounds like as if we are in Mars, but in actual sense, there are some clinical trials that have been done in terms of gene therapy for HIV, and mostly they've looked at use of CRISPR being delivered by an AAV vector, and this would be targeting HIV proviral DNA for excision of the virus, and if the virus is excised, then it cannot affect other cells, so you're clear. Then they've also looked at knocking out the CCR5 receptor. There they are mimicking what we saw with the BMT using donor cells that had the CCR5 mutation. Then they've also looked at boosting CD4 T cells in order to have a more robust anti-HIV immune response. And also they've looked at CAR T cell therapy for HIV. And on the arm for sickle cell disease, we know that there are already 2 products on the market that are based on gene therapy to treat sickle cell disease. So when we talk about gene therapy, we have to have some balanced optimism, because the safety profile is something that is continuously evolving. The biggest concern for gene and cell therapies is off-target effects. And here we mitigate this using high fidelity nucleases like the CRISPR, and also having a well-designed guide RNA design, because when you have a well-guided RNA design, then the CRISPR will be very specific where it cuts. Then we also have to worry about immune responses to the different delivery tools. For example, I've already talked about AAV, even the lentivirus itself. There is a possibility that the hosts or the individual that is receiving these gene therapies may elicit an immune response against them. So that's why there is a shift from viral vectors towards things like lipid nanoparticles or gold nanoparticles in order to avoid that immune response. But also to mitigate that, you can pre-screen these individuals so that you use the most appropriate delivery mechanism for them, or you use short-term immunosuppression during therapy so that they are able to take it up and not react to it. Then there is always a question of what are the long-term effects on one's genetics when you use gene editing tools. So here it's preferable to use non-integrating vectors like the AAV or plasmids. And then there is need to do clonal monitoring before reinfusion and long-term follow-up. At the moment, anyone that is in a gene therapy trial is followed up to 15 years at minimum. And that way we can catch any side effects, any changes in genomic stability early on, and be able to put in place mitigating factors. Then another thing that you would have to consider in terms of safety is the stress on the cells. So for gene therapy, we've said you need conditioning. There is vector for you to be able to infuse these cells back into the patients. Then the editing processes, and there is potential damage to cells and tissues. So there is need to optimize the doses and delivery conditions, and also use safer platforms to deliver these genetic modifications. So we said earlier that latency is the biggest hindrance to an HIV cure. So under gene therapy, how can we be able to reach those latent reservoirs in order to achieve the cure? So one of the ways is to engineer the vectors in such a way that they can cross the blood-brain barrier, because the neural system is protected from the periphery. So you can engineer these viruses in order for them to cross the blood-brain barriers. Then also for nanoparticles, you can make them functionalized in order for them to go to target cells. For example, if you want them to go to stem cells, you would put an anti-CD34 cell, an anti-CD34 antibody, so that they go towards just the stem cells. Then we also have cell-based therapy, whereby you do ex vivo editing of the stem cells or the T cells, and then you give these back to the individual. That way, you minimize any off targets, and the cells can home to the different organs in a natural physiology. Then we've also looked at direct in vivo CRISPR delivery. Here you target the viral or non-viral delivery of editing machinery directly to the tissues. The problem here is tissue penetration, avoiding off targets, and immune clearance. So this is work that is ongoing in order for us to see how to make it better. So just a reminder that we are coming from bone marrow transplants into ex vivo stem cell editing, and then finally in vivo stem cell editing. So the question is, with gene therapy, can it be scalable with where we've come from? So the potential for in vivo gene editing or gene therapy is that it has a very high potential for scalability. So here you can use one batch for many patients, unlike the ex vivo, whereby it is that entire cycle of removing. So you have to deal with one patient at a time to process this. Then we postulate that the cost of in vivo gene editing will be much lower than ex vivo gene editing once we scale up. And the infrastructure will be less specialized than for ex vivo gene editing, whereby you need an ISO class 5 laboratory, clean rooms in order to do that. Then for ex vivo gene editing, there is higher control in terms of safety, and the cells have to be quality checked, as we went through that long list. So that makes the cost of manufacturing ex vivo gene therapies much higher than for in vivo. Although the question would be, if you put these delivery tools in a syringe, what do they do? Where do they go? So there is systemic exposure. So it's harder to control what would happen. But the time from the time to actually treat will be shorter, because we are looking at single infusions versus the weeks that are needed from harvesting to editing, expanding, and reinfusing into cells. Then there are also immune challenges that you would have to consider. Systemic immune responses are most likely going to occur with in vivo gene editing, especially if it isn't targeted. And these may affect effectiveness of the gene therapy. Yet with ex vivo gene editing, this is limited because the cells are manipulated outside the body and then returned. So you'll have less immune challenges. So finally, what do we see as the roadmap to a one-shot HIV cure? If we can come from the current ex vivo gene therapy trials, whereby we are knocking out CCR5 or knocking out or excising the virus, and then moving towards in vivo gene editing using CRISPR and all viral vectors, then we can move towards clearance of the latency reservoirs. And in the nearest future, we hope that we shall be able to have a one-shot curative, single-day outpatient anti-HIV shot. So with that, I would love to thank all our funders and the support we've received over time. Thank you.
Thank you, Dr. Lois. Thank you very much for that very scientific but yet enlightening talk on gene therapy. Our dear listeners, viewers, colleagues, I'll just remind you that Dr. Lewis has been sharing on gene therapy, a cure beyond bone marrow transplant. Just before we go into our Q&A, I would like to notify you of just two things. There will be an evaluation form. Please take your time, do accord us a few minutes to complete it. Together we can learn more and improve this program. And it will look exactly like that. It's just a few questions. Please do fill it up and do share it with the friends that have not been able to log in today that you know were part of this program from the time we started. That is 8 webinars ago. So Dr. Lois, as you know, such scientific talks do not leave you without questions. And here I have been given 5 questions to share with you, and we hope we can get a response in addition to the ones that you'll see in the chat. Are you comfortable with 2, 3, or do I give you all?
One at a time.
Well, thank you. With the current economy and what is going on in the gene therapy space, what is the cost of receiving a gene therapy as an individual? And what is the cost, the estimated cost of starting gene therapy for a country like Uganda?
Thank you, Dr. Diana, for that question. So currently, gene therapies, for example, the ones for sickle cell disease, are very expensive. They are in about 2 million USD. That is US dollars. But there are others that are as low as 400,000 US dollars. So for a Ugandan or someone in Tanzania, or even in the developed countries, that is quite a very high price. But there are many factors that come into play. There is a lot of investment from the research and development process that affects the pricing of these therapies. So as a person in this field, I'm also an advocate for access to cell and gene therapies. So we lobby and will request for ways to make it cheaper within countries that have high burden of disease and actually need these therapies. Okay, so we lobby in terms of, can we get licenses to these the intellectual property or patents that led to these gene therapies, so that we can do local manufacturing. But we also look at it in terms of research, and we are like, how do we cut the manufacturing process? How do we make it cost effective? Do we produce our own lentiviral vectors or AAV vectors locally? Do we explore using no conditioning regimens in order to do these gene therapies? So there is a lot of work that is being done in order to cut the cost of manufacturing, which would translate into a cheaper therapy that can actually be taken up by most of the individuals in low and middle income countries where most of the burden for disease is. Yes. But also in terms of delivery mechanisms, if we switch from lentiviral vectors or AAV, to things like nanoparticles, lipid nanoparticles, then it will be much cheaper to achieve this. Thank you.
Thank you, Dr. Lois. And the next question has been partly answered by what you just talked about, which is geographical access. What is the future of having gene therapy in Uganda? Both HIV and sickle cell.
That future should not be a one-man a one-man job. For us to have gene and cell therapy within Uganda, within Tanzania, within Kenya, or anywhere on the African continent, we need to build capacity. That is having more individuals who are trained in different aspects that feed into gene therapy, from molecular biology to immunology to physicians. Then also, apart from that capacity, we need to get buy-in from our own government, because this is, African problems should be solved by Africans. So our governments need to put some investment in not only research, but also in development of new science, innovative solutions to the burden of disease. So we've started since GGTI was formed. GGTI is the Global Gene Therapy Initiative that was formed in 2020. So we've made some headway in the gene therapy field. Our colleagues in Muhimbili, they have a bone marrow transplant unit. That is a good start. And now they are moving towards research. So we believe that the future is near or here. Yes, thank you.
Thank you. And as you can see from the chat, question 5 relates to the same. And what are we doing as Ugandans to get to reach this goal?
Yes, as I've already said, as Ugandans, we are training more people. We are doing awareness and education about gene therapy, because when you do not have community, when you do not have regulators, when you do not have people within healthcare workers understanding what gene therapy is, then you cannot get the quorum to move forward, right? So just like this webinar, we are doing education for the community. We are doing training capacity for health workers and basic scientists to do this work. And we are also, we've made collaborations to get access to these gene therapies. And now we are working towards infrastructure so that we are able to do the first clinical trial on cell and gene therapy in Africa. We've also engaged with our regulatory bodies. There has been, the WHO has guidelines, there are FDA guidelines for gene and cell therapies. And now we are working as Ugandans to have these regulations in place, these policies that support this kind of work in Uganda. Yes.
Okay, thank you. Dr. Lois, from last year when we had the first gene and cell therapy symposium, this question has been coming up and has been on almost all the webinars. Please clarify to us again. In the case of sickle cell disease, after receiving gene therapy and they married a carrier, would they have normal children?
Oh, if you are a person living with sickle cell disease and you've had gene therapy and you marry a carrier, would you have
a normal child?
A normal child.
I think normal here refers to either having a configuration of AA or AS.
So by virtue of you getting gene therapy, we do not change the germline. So this gene editing is in the somatic cells. That is all the cells of the body except the spermatozoa and also the ovaries, like we do not touch that. So we just change the cells that make the blood, right? And that is stem cells. So for that instance, that means even if you get gene therapy, if you get married to a carrier and have children, they will have sickle cell disease. So it isn't an this treatment is for that individual. It cannot be inherited to the next generation. Yes.
True. And following on that response, I would like to implore all of us, all listeners, to be advocates, to test, screen. Because indeed, we do not inherit gene products. We do not pass on gene products. So in making decisions for birthing, we need to make wise and healthier decisions, and that would mean we make intentional decisions about who we marry. And if we choose to marry a carrier in this case, we have to make a decision and a follow-on decision of whether or not to have a child. If to have a child, we can use advanced scientific methods to have a healthier child. There are more questions coming for you, Dr. Lois. More scientific in the area of clinical translation, picking from your Bible. You talked about people who receive BMT having to take lifelong immunosuppressants. Question is, what would happen if they did not take them?
So when people receive BMT, there is need to do immunosuppression. This immunosuppression is for the person who has received the BMT, not to reject the transplant. And even if it was a kidney transplant, even if it was a liver transplant, you would be on these immunosuppressants for long. And when they stop taking them, we've seen in literature, even if it's after 5 years, there is usually rejection of that transplant, and therefore you reverse what you had done before. However, there's been some research whereby people get donations from closely related donors, and but this, let me say, you get a kidney transplant, but also stem cells from that donor. And so this mix of your stem cells and donor stem cells can kind of help to mitigate rejection. And these people have been able to get off immunosuppressants after like 3 years. However, this is ongoing research. At the moment, the standard is that when you get a transplant, you need to be on lifelong immunosuppressants to avoid that rejection.
Okay, thank you. You have some more questions about the reservoir that's in reference to HIV. I'm sorry, I'm going to swing you from sickle cell disease to HIV, given that they are the two strongest for us in Uganda as potential recipients for gene therapy. You earlier shared with us that when we receive gene therapy, it pushes the virus into latency that was under block and lock. What is the risk? Maybe there is no risk, you can clarify to us. What is the risk of the reemergence of infection? Given that after receiving gene therapy, we will be put on immunosuppressants for a while. What is the risk of having viral reemergence?
So no, just to clarify, when someone receives gene therapy, they are not on immunosuppressants for a while because usually it's a self-donation. That means, Dr. D, you your cells are collected from you, right? Taken to a laboratory, do the gene editing, and your very same cells are put back, except that now they are protected against HIV. With block and lock, you're giving drugs. These are drug molecules. Excuse me. Whereby they would make the virus go into deep latency. And therefore, it cannot replicate. And until the point when these cells naturally die off, then that virus is also eliminated through that cell death. And if it's a gradual process, it won't happen in one go. Eventually, those infected cells would all die off. And because now the new cells are no longer being infected, then you're cleared of HIV. Now, there is a risk of what we call viral escape. So with all these strategies, if you're using latency reversal agents or latency activating agents, then that means that that is drug pressure. The virus has been known to evolve. So there is a likelihood that with those strategies, if we use them singularly, that the virus is likely to evolve and escape from this lock that you've put around the cell, okay? And therefore, we postulate that it will be best for such strategies to be done in combination. You do block and lock, but you also put antiretroviral therapy. Maybe the dosing will be lower, but it is in place. Or you do block and lock, but you also boost the natural immune responses of the T cells, such that in case of any viral escape, it can be cleared by these super CD4 T cells. So looking at it, the most likely way to achieve this HIV cure without viral escape is having a multi-pronged approach, even if you're using gene therapy as a cure. Yes.
Thank you. And I think that responds to Charles Brown's question of any other ways to detect and initiate treatments to control the reservoir.
Apart from early detection,
I believe the use of boosted molecules will also control the reservoir.
Yes.
And there was another question actually in relation to that. How do you monitor the reservoir and all its challenges when we use gene therapy?
So currently there are some diagnostic tools for monitoring the HIV latency reservoir. They are complex, laborious, highly specialized, so it is so would apply the same, like what we call the IPDA test assays. But they also have their demerits. They are not quite efficient at measuring the reservoir size. And that's why sometimes, despite ART being in place, you find that there are blips still showing up. So there is still work to be done in the aspect of monitoring or even measuring the size of the latency reservoir.
Thank you. Dr. Lois, as you can see, there's a lot of interest on access of gene therapy in Uganda.
Mm-hmm.
Probably I'll request that you respond to that in just one statement. Is gene therapy available in Uganda? If yes, is it ex vivo or in vivo? If no, where can we find it? And what are the funding opportunities? About 4 questions
in one. So currently, gene therapy is not available in Uganda. For one to access gene therapy, if they're in Uganda, it would be a question of, can we link them to current clinical trials that are happening elsewhere. So that would be the way to access that. For sickle cell disease, gene therapy has been FDA approved. It's available in the US and UK. I think also Europe. Not so certain about that. Yeah, for HIV cure, this is still clinical trials. So the only way you can access it would be through a clinical trial. What we are doing as JCRC is that we've approached collaborators to allow us to replicate some of these clinical trials within the JCRC. And when they are available and it's time for recruitment, we'll let you know so that people can access it, at least initially through a clinical trial. Yes.
Okay, thank you very much.
And whether it's ex vivo or in vivo, ex vivo, there is no in vivo work that has gone to the market yet. And even the clinical trials are still in the very early stages for in vivo gene therapy.
Thank you. And there was actually a question related to the timelines for the in vivo therapy as a one-time shot.
As a one-time shot.
Any projection on timelines?
Do I give false hope or true hope?
Truth.
Truth.
Yes. Science is based on facts.
Science is based on facts. Okay, so currently we have phase 1, phase 2 trials. Phase 1 clinical trials for in vivo gene therapy for HIV. 5 to 10 years? I'm not sure. 5 to 10 years is what is my estimate. Yes, for the in vivo shot.
5 is not bad.
5 isn't bad. Okay.
Yes. Just a follow on for Robert about the person living with sickle cell that is with you. In the meantime, before access to gene therapy, I would encourage you to make sure this person is optimized for care. So do please find a good center, JCRC being one of the best that I know of, to have this patient optimized so they have more comfortable life, more meaningful life, while we hope for the gene therapy to be made available. Dr. Lois, please do share with us on the concept of ISO certification, how it makes meaning for us as Ugandan providers, Ugandan potential recipients. Do we have ISO certification? What are the processes? What is the infrastructure required? If it is there or if it is not there, and how else we can be part of this partnership to make it happen?
So in terms of gene therapy, when you look at if we are doing ex vivo work, ex vivo gene therapy, we will definitely need the ISO certification. However, with in vivo, that is for the manufacturing laboratory. Okay, there are standards that have to be met. So definitely ISO certification will be required. Current standards that are set is that if you're manufacturing ex vivo gene therapies, you need to have ISO class 5 laboratories. These are very clean laboratories. And the reason for that is safety. The main reason for that is safety. When you do in vivo gene therapies, okay, yes, you can maintain ISO classification, but it wouldn't need to be as stringent as for ex vivo, because here you would only be delivering the delivery tools in a single shot. Okay, so there is no exposure of an individual's blood cells or liver cells, whatever kind of cells, to the outside. So that kind of environment changes in terms of ISO certification. Yes.
Thank you very much. The follow-on questions related to care of persons with sickle cell disease. Immaculate asked, are there facilities that carry out tests that you mentioned? I will answer for Dr. Lois. Most of the tests are available in almost all facilities. She talked of organ function tests, the basic follow-up tests that we do for the warriors. They are available in all the centers that care for warriors. However, the donor match that are for BMT are what are unique. But for gene therapy, we do not need donor tests because this is self-donation. So all tests that need to be done are for the individual that is actually going to donate to themselves. Another question for you, Dr. Lois. Wow. From Adrian, given the different ex vivo approaches of the approved therapies, Casgevy and Lyfgenia, are there sickle cell patients better suited for the gene addition versus gene modification approach based on their biology? Number 2, is an anti-sickling electrophoresis type, HBA, better for some patients versus reactivating the switch to increase fetal hemoglobin?
That is a tough one. That's a tough one.
Maybe what our partners or viewers need to know is that gene therapy, like you said, is a self-donation. So there will be no circumstance when somebody with an HBA type will be self-donated, because that defies the background reason for the gene therapy in a person with a physical cell.
Well, I think
Dr. Lois will add on to the science of gene therapy.
Yes. Well, I think Adrian is saying when you do the Lyfgenia, which is gene addition, you're getting the adult HBA, right? In order to treat that sickle cell. Yet others who are getting the Casgevy are getting one which reactivates the fetal hemoglobin. I don't know who would be better suited for what. The clinical aspects, I don't know what would be better for who. And then also in terms of HBA being versus HBF, biologically, I would say HBA would be better than HBF because there is a reason for the switch. It means that the A is quite more efficient versus the F. That's why we switch, naturally from F to A. But any data to back that up, I do not have it. So we shall delve deeper into that. Yeah.
Thank you. There's another question for you, Dr. Lois, that compares the two therapies. What are the indications to do gene therapy in HIV-infected persons? Part B to the same question is autologous or allogeneic BMT in HIV patients. Which one achieves better cure?
So for HIV for people living with HIV to get gene therapy, currently it would be with viral suppression, no comorbidities that would affect uptake of this therapy. Autologous would be better to achieve a cure because there is no risk of graft versus host disease. Therefore, if you do a self-donation, then that means that is eliminated and there wouldn't be need for immunosuppression. So autologous would be best for HIV cure rather than the BMT. Because BMT, for that chimerism, for us to say, okay, this is good enough engraftment of the donor cells, and for them to be functional, it would take time. So an autologous transplant would be best.
Thank you. Once again, there's a question related to capacity. When will the Ugandan community be independent enough to manufacture gene therapy?
Okay, that is interesting. Well, I'll pose back the question to everyone. As I said, this isn't a one-man goal, okay? So we have scientists from different spheres that can work towards this. And so all of us, let us try to find solutions based on our training let us all work towards a solution to the disease burden that we are seeing in our population. So molecular biologists, can we find new targets for an HIV cure? Can we get new delivery tools that are more efficient? Can we get editing tools that are more efficient? Immunologists, what do you see that is special about elite controllers that we can translate into a cure? Clinicians, what do you see in terms of conditioning regimens that can make this process much cheaper and much realistic so that it can come to Uganda? So that is a ball that I throw back to everyone in the audience, because it's a joint effort, it's a collaboration for us to find the answers to this.
Thank you, Dr. Lois. Our dear viewers, listeners, a big round of applause to Dr. Lois. I will apologize on Dr. Lois's behalf and on my own behalf in case there are questions we've not responded to, but we hope we shall keep in touch with all of you with the live interactions on YouTube, on Zoom, to respond to all of them today. Otherwise, I thank you all for being part of the session. I thank the EDCTP project for helping us walk through gaining knowledge and gaining more and more. Hopefully, we shall be awarded certificates and diplomas by the end of this program. Our next webinar will be on the 8th of December. It will end with a Christmas party for those that will be in attendance. A big thank you to EU, our funders, our partners in this journey and growth. Wish you all the best in the coming days, and until we meet again on 8th December, wish you well. Keep healthy, keep happy. Thank you.